My name is Claire Finkelstein, and I'm the faculty director of the Center for Ethics and the Rule of Law. We are delighted to be here in the Annenberg Public Policy Center. It has been my great honor and the honor of the center to be able to partner with the Annenberg Public Policy Center and Kathleen Hall Jamieson, who is over here. We are absolutely delighted to be here and to be co-sponsoring this conference with them. As you know, the Annenberg Public Policy Center has been a leading voice on the science of science communication. And there is no field in which there is more need for clear communication than on the subject of the COVID origins debate. This is a critical topic on which there has been nothing but fighting, confusion, mystification, and so we decided to get 5 of the smartest, most interesting, and level-headed people that we could find together to rise above the fray, to have a serious, intelligent discussion across a rather great divide. Every day you pick up the paper, there's some other article, even within a single publication. I'm sure many of you read the New York Times, and if you're like me, knowing rather little about this subject, though I must say, after weeks of preparing for this panel, I know quite a bit more than I once did. Pick up the New York Times and you end up with whiplash pretty quickly, because one day you read an article saying it's the market, and the next day you read an article saying it's the lab, and you're wondering what'll be next. So I really thought it was important, especially important, having this conversation in a university where we have premier researchers into coronavirus and coronavirus origins, with others working in this space across a variety of sectors, to have this conversation in a serious way. And hopefully we will set an example for others so that we will start to enlarge the public policy discussion, start to have national security professionals, journalists, scientists of all different types, and public officials start to have a clear, intelligent conversation about a critical topic. I don't need to tell you how important getting this right is, not just for being clear about our recent and deadly past, but to be clear for the future and for preventing the next pandemic. So that's what we're going to hope to contribute to in an intelligent way today. So let me go ahead and introduce our panelists. To my far left, we have Dr. Sergey Pond, who is professor of biology at the Institute for Genomics and Evolutionary Medicine at Temple University. Over the past 20 years, Sergey's group has developed and applied sequence analysis, bioinformatics, and statistical methods to the study of viral pathogens. His research tries to identify and implement a practically useful and statistically justifiable solution to particular problems, and focuses on delivering high-quality, computationally efficient, and user-friendly scientific software. They have published 20 or so papers in this area, and additional areas since the start of the pandemic have made a tremendous contribution. Second, to my left still, we have Dr. Jamie Metzl, who is the founder and chair of the global social movement, One Shared World, a senior fellow of the Atlantic Council, a faculty member of Singularity University and NextMed Health, and the author of 5 books, including the recent bestseller, Hacking Darwin, Genetic Engineering and the Future of Humanity. His next book, The Great Biohack, Recasting Life in an Age of Revolutionary Technology, will be published in May of 2024. In 2019, Jamie was appointed to the World Health Organization Expert Advisory Committee on Human Genome Editing, called the original COVID-19 whistleblower for his leading role raising the possibility of a research-related pandemic origin and advocating for a full investigation. He was the lead witness in the first congressional hearings on this topic in March of 2023. Jamie previously served in the U.S. National Security Council, State Department, and Senate Foreign Relations Committee, and with the United Nations in Cambodia, and appears regularly on national and international media. Next, we have my colleague, Dr. Susan Weiss, who is currently professor and vice chair of the Department of Microbiology and co-director of the Penn Center for Research on Coronaviruses and Other Emerging Pathogens at the Perelman School of Medicine at the University of Pennsylvania. She previously served as Associate Dean for Biomedical Postdoc Programs from 2010 to 2019. She has worked on many aspects of coronavirus replication and pathogenesis over the last 40 years, making contributions to understanding the basic biology as well as organ tropism and virulence. This work included the murine coronavirus infection of the central nervous system disease, a mouse model of multiple sclerosis. More recently, she has worked on MERS-CoV and since 2020 also on SARS-CoV-2. Her work for the last 10 years has focused on coronavirus interaction with the host innate immune response and viral innate antagonists of double-stranded RNA-induced antiviral pathways. Just this week, I'm proud to say it was announced that Susan was elected to the National Academy of Sciences. Congratulations, Sue. To my right, Dr. Gigi Quick-Grunwald, an associate professor in the Department of Environmental Health and Engineering at the Johns Hopkins Bloomberg School of Public Health, and a senior scholar at the Johns Hopkins Center for Health Security. During the COVID-19 pandemic, she led the center's efforts to track the development and marketing of molecular and antigen tests and serology tests, as well as the development of national strategies for COVID-19 serology antibody tests and SARS-CoV-2 serosurveys in the United States. She also has written about the scientific response to the COVID-19 pandemic, the contested origin of SARS-CoV-2, and the implications for national and international security. Dr. Grunwald is a member of the International Security Advisory Board, which provides advice to the Undersecretary for Arms Control and International Security at the Department of State, and a life member of the Council on Foreign Relations. And last but not least, my own sister, Catherine Eben, who is a Vanity Fair contributing editor and an Andrew Carnegie Fellow. Her reporting on generic drug fraud, gun trafficking, coercive CIA interrogation, and COVID-19's origins has won international attention and numerous awards, including from the National Association of Science Writers, the Overseas Press Club, Investigative Reporters and Editors, and Newswomen's Club of New York. Her 2019 book, Bottle of Lies, The Inside Story of the Generic Drug Boom, was a New York Times bestseller. Her previous book, Dangerous Doses, A True Story of Cops, Counterfeiters, and the Contamination of America's Drug Supply, published in 2005, tells the story of how counterfeit drugs infiltrated America's pharmaceutical supply. Please join me in welcoming our esteemed panelists. So I apologize, I'm going to have to share the mic here with Gigi. My apologies to you, Gigi. So we sort of go back and forth here. Okay. Yours is dead also?
We'll share. It's a good...
Right. We're all going to have to share. We're all going to have to share.
Might just be off.
All right, well, we're working on that. I'll tee us up. So there's been a fierce debate about the origins of the pandemic. We're going to start by talking about this debate in a very focused way, and then we're going to draw the lens out and think about some of the implications of this debate. And we're going to move in concentric circles out to a sort of broader and broader look finally, looking at international implications and what we need to think about when balancing ethical concerns and national security concerns, which is where the specialty of the Center for Ethics and the Rule of Law lies, with the need to make advancement in science. How do we think about that? What are the national and international structures in place to help us navigate these difficult waters? And how do we make sure to advance the conversation as we work our way forwards and lessons learned from the current discussion? But we're going to start from the very narrow and most contentious part of the discussion, which is the debate about COVID origins. I would say that there are roughly 3 camps, and whatever language I use to describe these camps, someone on the panel, maybe everyone on the panel, will be unhappy with the description that I use. I'm not a scientist, so I'm inevitably going to say this wrong, but the 3 camps are roughly zoonotic origin, lab incident origin, and third camp, it doesn't matter, let's not talk about it. Well, I would say of that third camp, most of the folks on this panel are not of that camp. So we're gonna stick in the first 2 camps. There have also been some wild theories circulating on the internet and in other places, like it's a bioweapon. And what I like about this panel is that while the differences may run deep among our panelists, they're all within the range of reason, and we can have a sensible discussion among the sort of 2 prevailing theories in the mainstream press and mainstream literature on this. So I'm going to start, as we start to approach this topic, by asking Susan, who is our real coronavirus researcher and expert, just to give us a little primer on what is the history of coronavirus research and how should we think about the sort of place that we have reached around talking about the evidence around.
Okay, so coronaviruses, can you hear me okay? This is working,
Yep.
I think. Okay, coronaviruses are a really large group of viruses that infect just about every species of animal. So they've been of real important pathogens for cows and pigs and birds and other wild domestic animals, and there's been a lot of vaccine research on these animals way back before the 60s and 70s even. So they're very prevalent in animals. But the first reports of human coronaviruses were back in the 60s and 70s, when they were known to be cold viruses, caused the common cold, OC43, 229E. You might have heard of them now in the literature, I mean in the newspaper even. So, but they were cold viruses, so people weren't doing so much research on them. So I got involved in these viruses back in 1980. So from that time until 2003, when we had the first SARS epidemic in southern China, there was a lot of research done by really a relatively small group of people. We coronavirologists were kind of not in the mainstream of virology, but there's a bunch of us working on just trying to understand these really interesting pathogens, mostly studying on the animal viruses because they were easy to work with and because they caused some very interesting disease models. So in 2003, I think everyone agrees now that the first SARS virus that emerged in southern China came immediately from a civet, a wild animal in a market, to a human, and that civet had been infected by a bat. So we learned then that that bats are full of coronaviruses and other viruses as well. Ebola comes from a bat. Many viruses come from bats. And it's a really interesting field of why bats carry viruses and don't get very sick from them. Anyway, so after the first SARS epidemic, things were kind of quiet in the coronavirus world. There were more human viruses discovered, NL63, 229E, also causing more mild upper respiratory diseases. But SARS taught us that coronaviruses can be lethal. And in fact, we researchers were shocked at that time that it was actually a coronavirus, because till that date, we only knew about cold viruses, even though we knew that animal viruses could be quite lethal. I'll get there. So then we had MERS coronavirus, which another in 2012 emerged in Saudi Arabia. It was transmitted from a bat to a camel to humans. So again, we have that same zoonotic transfer. Then again, so we should have maybe been thinking more realistically about these viruses as pathogens. And then in 2019, as we all know, the third lethal coronavirus emerged in a different part of China. I personally think it almost definitely came from a bat and was transmitted to humans, probably through a wild animal in the market.
Sorry. So for you, Susan, who have seen and who have traced other coronaviruses and traced the history of these viruses over many years, there's a familiar pattern. They all or almost all come from bats?
I should say also not all from bats, but some from like rodents. But even the non-lethal ones, OC43, the cold viruses, can also be traced as transmission from animals. From, like, for example, OC43 would probably came from cows.
Right.
And they're very closely related to cow viruses.
So Sergey, why doesn't everyone just assume, okay, the current SARS-CoV-2 virus that caused the most recent pandemic came from a bat, and we just have to find the intermediate animal?
Thanks for asking this question. I think it's a great one. It did come from a bat, but what it made, which stops it made along the way, is what we're really looking at. We do not get to observe patient zero. That only happens in the movies. We get to observe indirect evidence of where the virus came from and how it might have transmitted. And what it's important to appreciate is that in even well-established diseases like HIV or various recent outbreaks of different influenza variants, we don't ask questions to the same degree of precision. We do not want to know exactly what spot it came from. We're content with much more broad questions. What's the host animal? What's the range of dates where it came from? The way we ask these questions is we look at viral genomes. Viruses are very interesting to study as evolving organisms because they belong to what we call measurably evolving organisms. You really can see that. If you're infected with coronavirus, it evolves within you. Within a couple of days or weeks, it will acquire some mutations. It's not going to remain constant. So it leaves a trace in its genomes by collecting viruses from humans and comparing it to animal viruses that could be potential hosts. We can estimate, and the important part is estimate. This is not measuring. This is all based on fairly complicated statistical and computational models that are becoming progressively more complicated over time for good reasons. But we can estimate that at best, the closest animal isolate we have to SARS-CoV-2 is about 40 years, give or take, of evolutionary time that we don't know where the virus was. So that is sort of where we're coming from. And it could have done lots of different things in those 40 years, but that's sort of where genetics really gets us in terms of the closest isolates that we have now. So closest isolates would be from bats, as Susan pointed out. The other interesting thing that I'd just like to mention is that coronaviruses are truly ancient viruses. They're incredibly diverse. When we looked at their genetic data back in 2013, we basically estimated that they're as old as placental mammals, so 50 million years ago. They've basically been with us since we were here, since our ancestors were here. So they had plenty of time to adapt and switch around. So they're kind of very, very old viral family.
So what's the bottom line as far as you're concerned on the evidence of where the virus that caused the pandemic, SARS-CoV-2, came from? How do we, what evidence do we have? You're sort of saying that we just don't really have very much evidence.
So let me come at it this way. In preparing for this panel or thinking about our approach, I actually asked my sixth grader, if I told you that the virus came from an animal and infected humans, what would be the first piece of proof you asked me for? He thought about it, said, an infected animal. Right? And at the moment, to the best of my knowledge, we do not have evidence of an animal at or around the wet market in Wuhan where one of the prevailing theories states the virus made the original jump to humans. It is not necessarily disqualifying, but it is an intuitive gap that for a common person that is not thinking about it deeply is a big gap that needs to be addressed.
Okay, great. So we'll get to audience questions towards the end, and we'll leave plenty of time for it. I'm going to ask Gigi now. So you very much disagree with this. Tell us what, in your mind, is the evidence for the market hypothesis? And excuse me for passing the mic back.
No worries. Can you hear me okay?
Yes.
Okay, so part of the issue here is that in 2003, after SARS-1, there was an immediate connection with the marketplace. And they went to the market, you were able to sample the animals, you had lots of fuzzy civet cats, staring at you. You were able to figure out that those were the problem. This is not what happened in 2019. We had cases that were apparently connected to the market. More than half of the original cases were connected to the marketplace. But on January 1, 2020, that market was cleared out by Chinese authorities. And so there were no animals left to be.
Can I just ask you to clarify before you continue? When you say the original cases were connected to the market, can you explain what you mean by that?
So people who had worked in the market or lived near the market or went to the market, more than 60% of the first cases of SARS-CoV-2 centered around the marketplace. And the fact that not all of them were, then, a larger analysis later that was done found that the cases were in the same geographic area, which was about 10 miles away from the laboratory. So there was like a distinct, statistically significant connection to the market versus the laboratory. But you didn't have the animals, and you will never have those animals there because they were all cleared out. There was environmental samples taken, so samples were taken of various stalls because it was a seafood market, and so you wonder, like, why do you have animals at a seafood market? But it turns out, fortuitously, a tick study that was a tick disease study that was done in the months prior to SARS-CoV-2 emerging, they found, they documented with photographs evidence of these animals and the prices, many of which were illegal. They're not supposed to be there. Treated in terrible condition, shoved into small cages into the marketplace. So we know that there were animals there by photographic evidence at some point prior to the pandemic. We know that there are cases that are connected to the pandemic. We also know that there were 2 lineages of SARS-CoV-2. So there were like 2 distinct versions of the virus that were spreading, and only one of them was the B version, ended up spreading around the globe. So the A version didn't pick up, but what this might say to an epidemiologist is like, okay, multiple spillover events were occurring because you had an infected population of animals. So you had animals passing it to each other. We always think of ourselves as being the most important things on the planet, but actually animals get sick too when they give each other diseases. And so, they were getting each other sick, and then that spilled over to people. And that's why you have these 2 lineages. There's also these environmental samples. So the ones that were positive for SARS-CoV-2 were associated with places where you'd expect animals to be. So the areas where these illegal animals were butchered, around the drains, and sadly, and I feel regretful that I now know what this is, a hair and feather removal machine. So that was also positive. So there was a recent, as the gentleman just referred to, data that the Chinese had withheld for 3 years about some other environmental sample data that was taken at that time. And in that data was a lot of raccoon DNA as well as other things. You're never going to be able to say, like, it was a raccoon dog, although it's a really cute animal when they're healthy, so I suggest you take a look at the pictures. but they're,
Was so tempted to put up a slide of raccoon dogs, but I
but
restrained
that's not what they looked like in the market.
Yeah.
Like these animals were really poorly treated and stressed. And so, we don't know exactly, what leads these diseases to be, spill over to people, but stressing animals out, treating them poorly, and stuffing them together in a place, in one place is not a good idea.
Thank you. Speak a little bit more into, you're saying raccoon dogs were known to harbor SARS-1?
Yeah.
Yeah.
Experimentally infected, so they're clearly a good suspect for a spillover.
So while we're on the topic of raccoon dogs, I'm going to ask Jamie to weigh in. Now, let me first just summarize. One of the recent whiplashes that you might have had was the New York Times published what seemed to be a breakthrough article, raccoon dog DNA found in close proximity to SARS-CoV-2 environmental samples, seeming to suggest that there may have been infected raccoon dogs at the market. That would be the intermediate host animal that would bridge the gap between the infected bat, right? So that would be the smoking raccoon dog, right? Except the next week, there was another article, and maybe, Jamie, you can explain
Yeah.
what the next iteration in that was.
Maybe I'll do that and then go a little broader. First, let me say I love animals. I love raccoon dogs. I'm against the wildlife trade. I think that we should end the encroachment into wild spaces and slow global warming. And I'm pro-vax and pro-Fauci and all the things that many people in this room probably share. But I also think that we need just to look plainly at the evidence and be as honest as possible with ourselves. And so let me start with the market origin hypothesis. There is a debate about when the first spillover event happened. The genetic evidence seems to suggest something like October, November of 2019. And if that's the case, then it would make just obvious that humans, which is what the Chinese scientists who actually did this research believe, that humans brought the SARS-CoV-2 virus to the market. I think that is, in my mind, it's a very strong case. And so that's one very important piece of this. You talked about this evidence. There was this story. First, it was in The Atlantic, and then it was in the New York Times. And the Atlantic article said, the strongest evidence yet of a market origin. That whole story has basically been entirely repudiated by the analysis of Jesse Bloom. There may be different views on this, and I respect that, which is why the New York Times looked at its coverage of that story and then retracted it. I mean, there are lots of reasons why we need to look at the market origin hypothesis. But in my view, and there are differences of opinion, and I respect that, but very, very senior scientists believe that it probably comes from the market, and equally senior scientists believe it probably doesn't come from the market. And that's why it's important for us to open our aperture a little bit and say, well, how do we know, how do we interrogate the question of pandemic origins? What are the kinds of ways that we can look? And certainly, given the paucity of data, we are all in many ways just trying to do our best analysis with very limited data that has been massively manipulated by the Chinese government. It's irrefutable that China has manipulated the type of data that's available. It's destroyed samples, hidden records, imprisoned Chinese citizen journalists for asking the most basic questions. It has prevented international investigations. It blocked the WHO from sending its investigators to Wuhan. And the list goes on and on and on. So if we say, well, what are the types of evidence that we can look? Certainly, we can and should do the genetic analysis of these small number of genome sequences that we're getting from the Chinese. And there are different interpretations of that, and that's fine. But we also need to look at the big picture. So what do we know? We know that we had an outbreak that happened in Wuhan, where they don't have the type of horseshoe bats in the wild that are the ancestral carriers, but as Sergey said, that was a long time before. What Wuhan does have is China's first level 4 virology institute with one of the world's largest collection of bat coronaviruses that was doing very aggressive research on these viruses. By their own admission, they were trying to create these chimeric viruses. And there are people who say, well, Susan, I think will say, because we talked about it before, well, this isn't possible. But these exact scientists at the Wuhan Institute of Virology thought it was possible to engineer SARS coronaviruses to make them better able to infect human cells. And that's why they were part of an application to the US Defense Department in 2018, seeking $14 million to do exactly that.
Okay, so let me put a pause here, because we've covered a lot of ground in your comments, Jamie, and I want to give Susan a chance to get in here. Let me just wrap up our discussion of raccoon dogs, that we could spend the whole rest of the time on raccoon dogs, but let me just, as I understand it, the Bloom analysis, and I'm going to ask you to comment on this, Susan, if you care to. The Bloom analysis that answered this initial data says not only did the data correctly interpreted not show that there were infected raccoon dogs at the market, but actually showed a negative correlation of SARS-CoV-2 with raccoon dogs, because in fact there was a lower concentration of SARS-CoV-2 in the raccoon dog samples than for other animals that were at the market. So the question is, what remains of there may be other evidence, but what remains of the raccoon dog hypothesis?
Gigi, do you want to comment on that? Because I should
Yeah.
I would like to comment on the manipulating the genomes in bat viruses, but let Gigi comment on that first.
Sure, I can comment about Jesse Bloom's comments, or his reanalysis of the data. I think that it's kind of missing the big picture, because the big picture is, why was the raccoon DNA there anyway, right? It seemed that they shouldn't have been there. They were illegal animals to have at that marketplace. Basically, the fact that the Chinese had withheld that data for several years, it indicates that they knew that that this data would show that these illegal animals were there, and they're where you'd expect them to be. And I think anything more than that is like, if it's connected to how much of the sample is connected to the SARS-CoV amounts in the sample, it's how many angels can dance on the head of a pin. Because the main message is, they shouldn't have been there. They were there. The Chinese sat on the data for 3 years. They should have released the data sooner. What other data are they not releasing?
So at least everybody, let me just say, at least that's one point. I'm going to take my wins where I can get them. I think that's one point everyone on this panel can agree on, is we'd like to see the Chinese government releasing data more readily, and that data on the market and on the animals, as well as data on what's going on in the lab, should be released. Okay, now, I promised Susan could reply,
I just want to talk, I mean.
and then I'm going to ask Sergey, and then I need to
Go back
come to Catherine here.
Because it's going to be about raccoon dogs, and then we'll go back.
Which, is it okay for me to?
Who's on raccoon dogs?
I'll do that.
Okay, Sergey on raccoon dogs, and then Susan on manufacturing viruses.
So I think an important point to clarify is that nobody would dispute that there was active SARS-CoV-2 transmission that occurred at the market early. The big debate, which is absolutely critical for the origin, is who was the source. Humans is one possibility, so somebody brought it there and spread it around. We're actually very good at transmitting this virus to other animals. We gave it to white-tailed deer, we gave it to minks. There are cases of transmission to house animals. So we're a good source of SARS-CoV-2. Now, how do you differentiate these two cases in the absence of an infected animal? So genetic data analysis, which may come back to later, but it's very important to demonstrate that there could have been an infected animal at the market. And Gigi's absolutely correct. We may never get these data, and you could argue that absence of evidence is not evidence of absence, but I think the reason the raccoon dog was so important in this debate is because people understand that we need to find evidence of an infected animal. My issue with the raccoon dog analysis is we have to appreciate there's peer-reviewed, published evidence of analyzing the same data where people, respected scientists, arrive at diametrically opposing conclusions. There's no consensus there. The Chinese that published the data and generated the data interpreted as it was human introduction. My biggest issue is that these data fundamentally contribute nothing to our understanding of the animal source. They were collected a month after the introduction of the virus. They're environmental samples, which means you can just think about it. I mean, you take a swab in your kitchen, you know, and see what was there. It might have been your cat, might have been your guests, fleas, who knows there. I mean, everything is there. There's no information about when they were there, whether they're together. If you find a virus that could have infected multiple species, you have no idea which species it came from. So the issue is that fundamentally these data are at best consistent with either explanation. They cannot contribute new information
I don't think so.
about it.
Okay, you have disagreement on this point. Just before I leave you, Sergey, I want you to explain one more thing, and then I'm going to ask
I
Susan.
want Gigi to answer him.
Oh, no, wait. No,
Okay.
Susan, I'm not going to let you off the hook, because I know you have
I
something
do
to
wanna
say here,
talk
too.
about it. No, I
Hang
do.
on. You've done important work on the timing of the earliest DNA samples and other biological evidence that we have. Can you just explain, again, you started to explain that, what the issue is with the timing. There's debate about what the earliest when the earliest samples are from and why it makes a difference when we date the earliest samples from.
Well, there are really two questions there. I'll answer one and maybe come back to the other, because you can look at biological evidence, which would be cases or documented infections. That's something that's intuitively clear. If you had a patient infected with SARS-CoV-2 in November, it means somewhere, hypothetically speaking, then the virus had to be earlier than November because the patient was already there. The second line of evidence, which is usually brought here because we have genetic data is all that's really been released and incontrovertibly from the Wuhan market or from Wuhan city. We use statistical models to run back the time and estimate when the origin, when the ancestor of the virus existed. And one other thing that is often lost in this debate is that SARS-CoV-2 generated a tremendous amount in scientific research. I looked up yesterday, there are 370,000 scientific papers in SARS-CoV-2. That is insane. Nobody could read them, nobody could read even papers on origin. On origin alone, there are about 5,000 papers if you do it like this. So nobody can claim that they know everything that's out there. However, if you just look at the question of taking sequences from viruses and using various statistical models to estimate when the ancestral virus existed, there's also a breadth of opinion from highly qualified people ranging from these data simply are not able to give us the precision of the answer that we want, to estimates that range by about 6 months, depending on who did the analysis, which sequences were included, which models were used. You can have a scientific and technical debate about it, but I think the main point is that there is a range of opinion in what you would consider sort of the gold standard of scientific evidence, which is a peer-reviewed publication. So there's no precise answer. And I think that's sort of, I'll end with this, is just his desire to have a very, very precise answer and just say that everybody agrees on it is simply not supportable out there in just peer-reviewed literature. I'm not even talking about, you know, internet research that people do in their own time.
All right, so I want to go, and there's so much to cover here, and we're never going to cover it all, but we have so many topics to cover that I want to go on to the next thing that Susan was about to speak about. Let me tee it up.
Okay.
Which is the issue of manipulated viruses. Now, the first thing that I want to make really crystal clear, and it's the main thing that I learned from starting to work in this area and study what these folks write and work on, is the issue of manipulation of a virus is not the same as the question of a lab leak. And here, I'm going to let Susan, and then I must turn to Catherine, who has been patiently waiting here by the sidelines while we have this more scientific debate, to start looking at some of the journalistic questions and what she's found through her reporting, and because this is where this really starts to come in. But in the minds of the public, what has surprised me is how much these two theses have been run together. The thought that the virus was manipulated in some way or enhanced or manufactured with the thought that there was a lab leak. Now, analytically, these are two separate questions. You can believe that they were manipulating and enhancing viruses in the Wuhan lab and not believe that it leaked, or you can believe that it leaked and it was not a manipulated virus. So let's make sure that we're really clear about that, and I'm going to ask
Okay.
Susan now to speak about the issue of manipulation.
Okay, so starting off, I just want to say.
Just raise your mic is too far.
Okay, I just want to say there is zero evidence of a virus of SARS-CoV-2 or a close relative of SARS-CoV-2 in any of the labs in China.
Can I ask, because I think this is a really important point. I think that's what I would call a faith-based argument, because we
Hey,
have,
let me
no, what I'm
Okay,
saying is,
hang on, wait a second.
we have
Yeah, I'm
no
gonna- let me-
clue
let me
what
let-
they had. Like, you
Can
would
I
have
talk?
to say, yeah, I'm sorry. My
Please?
apologies.
But all panelists need to raise their mics a little
All right.
closer to their mouths,
All right.
because we're having trouble
All right,
hearing.
so,
Okay.
so there's no documented evidence, I think that's true, of SARS-CoV-2 or a very similar virus in the Wuhan labs. There are millions of viruses in bats, right? So, and let's talk about manipulating viruses. Changing viruses, that is making recombinant viruses, moving their genes around, mutating their genes, is something that we do every day in virology labs. We do it. It's a very important tool that we use to understand how viruses work. We want to know how viruses infect cells, all kinds of things. And so what some people are calling like an evil experiment is taking part of a bat virus, the spike protein, the protein that we are all immunized against in the vaccine, and putting that into a different kind of a human virus and asking whether that new virus can now infect human cells. That's a very important experiment. We want to know whether bat viruses have the potential to infect humans without going through an intermediate species. So I'm just giving an example of the really good reasons why we do these things. Another reason why we do these things, I think is important, is when we're making drugs and testing drugs against SARS-CoV-2. We want to know what parts of the virus are going to change to make us resistant to that drug. So pharma companies they determine what part of the virus is going to mutate in response to be resistant to that drug, and they have to test that by making another genetically manipulated virus. So these are really important processes. And I want to emphasize too that we are not smart enough to know how to design a virus that acts like SARS-CoV-2. To take another virus that may not be that close to it, we would have to change it in a myriad of thousands of nucleotides. It's just so far away that we're not smart enough to do that. And even if we were to randomly change viruses and then ask, what's going to find the one that's going to kill people, we wouldn't know how to do that. You'd have to test it in humans. Like putting viruses in cell culture and passing them in human cells, selects for viruses that grow in human cells. Viruses are so adaptable, natural selection. They're going to keep growing in human cells. They'll be great, but then you put it out in the wild and they may be dead. And I know this from my own experience with manipulating mouse viruses, which nobody cares about when we manipulate mouse viruses. And I just want to say one more thing about, everyone's heard about the furin site. The furin site is a very short sequence in a spike protein that tells the cell to cut the protein there. That protein has to be cut to be activated. Furin sites, some viruses have these furin sites and they need them to enter cells. Some viruses enter cells very competently without a so-called furin site. So the idea that somebody might put a furin site in a virus and think maybe it'll make it more pathogenic, maybe not. It might kill the virus. It might change the structure of the spike protein that it's no longer infectious. So to me, it just doesn't make a lot of sense. And the Omicron virus, that's the one that spreads the best, right? It's been selected for spread. It has not a very good cleavage at its furin site. So we could say it evolved to actually be a less good virus if we think that the furin site is so important for pathogenicity. So all in all, I just see it's kind of magical thinking to think that people in the lab could create a monster virus like SARS-CoV-2. It's just doesn't make any sense from a coronavirologist's perspective.
As you've been educating me, Susan, since we've been talking about this, Mother Nature makes the best
They
viruses.
do, for sure.
So,
It's natural evolution.
Catherine, thank you for waiting so patiently. So you've been reporting on many different aspects of research, of both on the U.S. side and what you could glean on the Chinese side. One of the things that you reported on, not unique to you, this piece, is the 2018 DARPA research request to do exactly what Susan is saying would probably not be very efficacious. Can you tell me what you know about that research request to DARPA, why it was likely turned down, and what else you learned in the process of that particular, reporting?
First of all, it's just great to be on a panel with all you experts because I don't have to say too much, which is just wonderful.
Just raise your mic a little bit higher.
But what is striking to me about the 2018 grant, so this was a grant application submitted to DARPA, which is a DoD, Defense Department entity that does scientific research, and it was submitted by the lead coronavirus scientist at the Wuhan Institute of Virology, partnered with a scientific nonprofit based in New York called EcoHealth Alliance, as well as a North Carolina virologist. And what they had proposed to do back in 2018 was to take a wide range of bat coronavirus samples and manipulate them and possibly try to create cleavage sites in them to gauge the infectiousness of that insertion. A couple of things are really striking to me about this application, and one is when we found out about it. Because we did not find out about it until, I think, at least a year after the pandemic began. And I think it's significant because you would imagine that anybody involved in research like this would want to disclose this. So the fact that this wasn't disclosed immediately is sort of one of many omissions or things we learned about subsequently. And in fact, one of the scientists who was involved in the grant application was also involved in organizing an early letter in the Lancet to say we really abhor conspiracy theories that there is anybody could talk about a lab origin. So, one of the things that has been documented by journalists is a sort of an effort to create a narrative that basically took the lab leak off the table. I mean, I'm not saying that it is significant because I know or even have any certain idea or belief about where the origin came from, but just to say that when you look at the pattern of public disclosures and public omissions, that seems significant.
Well, can you explain a little bit why that happened? I mean, why do you think, so you've really drilled down in very, very detailed reporting on the relationship between U.S. agencies, more than one of them, and the Wuhan lab and some of the researchers that went to the Wuhan lab. A pandemic that killed 20 million people, you might think that we would be doing very extensive research, and moreover, that there might be more disclosure from our own intelligence agencies. Instead, we're in a very strange position right now of having two intelligence agencies that have come out and said that they believe it was the result of a lab incident of some sort. That's the Department of Energy and the FBI. And other intelligence agencies that either have no idea or believe the other way. And we have, a kind of bafflement from our own government. Why has this not been better investigated? Why do we not have more answers?
Can I just say something about
Let me invite Catherine to finish and answer the question, then I'll get other people in on this, okay? Thank you.
Well, I think maybe to just answer, you have a number of questions in there, so just to sort of address them, I'd like to talk a little bit about how I began reporting on this and what I found.
Go ahead.
In my first story. So really, my interest in this was sparked by something pretty mundane, which was just my editor and I putting our heads together and asking, what are the unanswered questions of the pandemic? And he said immediately, the question of origin. So I began doing some reporting. My background on this was that I had spent 10 years reporting on pharmaceutical manufacturing plants in India and China, and had uncovered an enormous amount of data fraud in those plants related to generic drug manufacturing. And that led to my book, Bottle of Lies. So what I understood from that reporting is that while there are very often universal standards and regulations, what goes on in many facilities around the world doesn't necessarily match the regulations. So there is this distance between what's on paper and what actually happens at certain facilities. So that was the my background as a journalist going into this. Once I began reporting, what became very clear to me is that there were non-crazy people inside the US government. I mean, there was, Donald Trump, who we have strong reason to doubt anything that comes out of his mouth. But there were, and he had said early on in April of 2020, it came from the lab and we know. And, that almost created like a sort of antibodies to even the idea of considering that possibility, because I think decent people do not want to align with an agenda that they view as, full of conspiracy theories, racism, and xenophobia. So I think that really led to many people in the mainstream media not really taking this seriously. So when I began my reporting on this, I was pretty surprised. I was like, where is everybody? I mean, we'd all competed as journalists throughout the pandemic to get to certain sources, to beat each other to stories, and suddenly, where was everybody? But, as I began reporting, it was clear there were non-crazy people inside the US government, inside the State Department, inside the National Security Council, who were raising very serious questions about this and trying to investigate it and being blocked. And their questions were being thwarted, and their queries were being, they just encountered one blockade after another. And what seemed clear from my reporting is that there were concerns within the sort of governmental scientific community and inside the NIH that there was some exposure for the government here, not because they thought they'd created the virus, but because they'd funded research at the Wuhan Institute of Virology. And they had promoted research, risky coronavirus research with Chinese scientists. So there was quite a bit of pushback from US scientific agencies into further looking at these possibilities.
So you believe there were conflicts of interest that stunted the investigations?
I do.
Is what you're saying?
Yes, I
Okay,
do.
Susan, I know you feel strongly on this. Let me let you in.
I want to say, Catherine said that's really not so.
It's hard to hear you.
Okay, this idea that they didn't reveal their this is not working, that they didn't reveal what was in their grant proposal is bizarre, because we write grants all the time. They're private, they're confidential, they're not public until if they're funded, they're public. But there's no reason why someone should go around telling you what's in their grant proposal. That's just not normal behavior. So there's nothing suspicious about the fact that we didn't know what was in their grant proposal.
Jamie.
There's a lot of issues, a lot of people, so we have to boom, boom, boom. So let me say just a few points referencing things that have been said. First, as a member of a community of people who've raised the possibility of a research-related incident or accident, there's a wide range of things it could be. It could be somebody from the Wuhan CDC going to a lab in southern China on a collection. We have videos of people without protective gear. There's a whole range of things, so I don't think we should be too narrow. With reference to what Susan said about the viruses that were documented to be held in these labs in China, we have no.
We're not documented.
No, some things we have no clue what viruses they had. Not all of them were even characterized. We have no clue what work was being done. And so that's why I called it, and my deep apologies for interrupting you, a faith-based argument. And I think it's really important in light of especially what I said before. And so as we look, for me, connected to what Catherine was saying about origins, I mean, in late January of 2020, it was pretty clear, looking at the evidence, that it could have been from zoonosis, and it could have been from a lab. And in my view, there was no possible analysis at that point that could say it was one absolutely or not, or the other absolutely. And that was why I just was personally insulted, and I thought it was wrong and just devious for people to say that anybody raising the possibility of a research-related incident of some kind to be a conspiracy theorist. And then when we look at the range of possibilities, I can see maybe it did come from the market. There's zero evidence of that to date, but there could be evidence, and I think we should be open to that. But are we going to say we're going to rule out the possibility that the people in Wuhan were doing the actual work that they applied for funding to do? I mean, it just seems like, and I was quoted by Catherine in one of her Vanity Fair pieces saying, if I applied for funding to paint Central Park purple, and tomorrow we woke up and Central Park was painted purple, I think it would be fair to for people to ask, hey, how did Central Park get purple? And I would expect a knock on my door.
So excuse me, I will invite the audience absolutely to comment and question when we get to that part. I think this is a good moment for us to ask a sort of meta question, because you can see on this panel, and this is a group of people that obviously disagree deeply with one another, but I'm delighted that they're willing to talk to one another, but there are folks out there who are absolutely not willing to talk to one another around this topic. And it's extraordinary to me, as someone who has run a national security and ethics center for over 10 years and who has dealt with the most contentious issue that this country faces. We dealt with the Snowden revelations, we dealt with the NSA spying on Americans that then became very controversial in the wake of those revelations, and I put spying in quotes. We literally had the second in command of the NSA in a room with the ACLU. We've dealt with the Trump investigations, and I have never had an issue that was as contentious as this one. And it's extraordinary because it's so hard to follow. So why are Americans so exercised about it? And why is it so hard to talk about it? And Gigi, you and I had a chat about that, and I thought you had some interesting thoughts about that, so I wanted to get you in on this.
Well, I think, I mean, it's this answer, this has a lot of ways I could take this, but I think, public health is by nature political. I think there was a time when people said, oh, this is just science, and so we don't need to get into politics, but from the beginning of public health, it has always been very political, and so I think acknowledging that the origin question was going to be a politically charged discussion right from the beginning is important. And Trump declaring it so definitely made it difficult. But that doesn't mean that things weren't considered. Now, I want to say you were a little light on the biosecurity elements of my background, but I mean, for many years have every time there is a pandemic, any kind of disease outbreak, there is a question of, is this deliberate? And that should be within every public health, responder's thought. And sometimes, sometimes that answer is more complicated, or so I think. I think it's not the case to say that people would have agreed that it could have been deliberate, even if or an accident, even though Trump said it, because people and scientists can be contrarian, and they can say their own thing. They don't vote as a block. They don't think as a block. In fact, they tend to be quite independent thinkers, as anyone who's probably tried to assemble a faculty meeting of scientists can attest. So I think that some of these questions are challenging, but this is not the direction that you want to go. Do you want to talk about?
No, well, so go ahead, Kathy.
I just wanted to clarify a little bit maybe what I meant, which is that, yes, while I meant it did create sort of an antibody against that consideration within the government, I really intended my remark to mean that it created that reaction within the media. And so that there was really, I think, much less consideration of this in the mainstream media than there would have been had Trump not made that announcement in April of 2020.
But some of these things, though, I mean, you'd swear that the only people getting infected by coronaviruses are scientists, when in fact, people get sick and die from diseases that we don't know what they are all the time, and the people who are, and we don't make a PhD case out of it, we don't figure it out. I mean, we have all gotten colds and we did not know what they were. So the question is, why wouldn't you have people trying to figure out what diseases bats have that could be spilling over to people? It's really important to figure out.
Yeah.
So to close out, and in fact on that topic, to close out this part of our discussion, and then I want to go to some broader themes before I invite the audience in, which is very soon.
Comment on his comments.
Okay,
about
quick, and then I have a
viruses.
question for you.
Okay, because I just want to comment about, this is what the scenario you said it includes fantasies, right? Because you need to make a virus, like, as I said before, you need to have a virus to start with. So you take viruses out of bats in bat guano, and they're mostly not viruses, they're pieces of RNA. They're not infectious, they're just junk, but they're sequences that you can sequence, right? So how do you get a virus? You'd have to find a real virus, and you'd have to isolate that virus, you'd have to grow it up, you'd have to copy it into DNA, you'd have to manipulate its genome, you'd have to be really lucky to get it just the right way to make it infectious for people, and then you'd have to figure out how to make it infect people, if it's going to infect people. It's just, the way you said it, it sounds very smooth and easy, but it's not that true.
Right, so actually,
it's not easy
Susan,
to make a virus in a lab.
my question to you really follows on that point, because you've been very strong in making the argument about manufactured viruses, the implausibility of it in your view, and so on.
It's possible. I'm not saying it's impossible.
But going back to the way I teed this up originally, that is very, very different from the lab leak hypothesis. So my question to you is, how open are you to the hypothesis that it could have been a result of a lab incident or a lab leak from a non-manufactured virus?
Okay, so I just wanted to answer that to Jamie's question. I think it's not likely. I think the epidemiology around the market is really strong. So that's sort of negative evidence that it came from the lab. I think that viruses, there have been, quote, lab leaks. SARS-1 infected a couple of people in the lab, but that's when they had a virus. They could grow up and make vats of it that you could get aerosols and people could get infected. If you have a little bat guano virus in a little petri dish in a tissue culture hood that's protected, or even if it's not that protected, you have a little bit of virus, it's really unlikely that it's going to get into a human being. It's possible. I'm not saying it's impossible. It just seems really unlikely to me. And there's no evidence for people being sick in the lab.
Can you just explain why it is unlikely? More unlikely, you're saying, in a lab where people are working with deadly viruses than in a market?
Oh, because
where
in the market,
animals
are breathing, and these are respiratory viruses, right? In a lab, it's in liquid, in a tissue culture dish.
But they had animals in the lab also.
Yeah, but we don't have the virus. So, but we're talking about lab leak. You think
we're talking about bat viruses
Then they would
in
have
a
to have
natural bat viruses, non-enhanced.
a natural bat virus will not likely infect a mouse or a guinea pig.
But could it infect a human?
Maybe, maybe not. But how is it going to get into that human? It doesn't fly. I mean, it'd have to be you'd have to inhale it, and you don't really inhale things from Petri dishes.
Okay,
It's just
so,
unlikely.
But they
So-
did have bats and animals.
so one second.
like
So,
bats.
one second. So, Catherine, you've reported
But they'd have to
that,
have the
one
virus.
second. You've reported that patents and procurements from the WIV that could indicate a response to a biosecurity incident in 2019. Can you talk about that?
Yeah. You know, we do know, because we have records of it, that the Wuhan Institute of Virology was taking a series of steps to improve biosecurity. But one of the things that they did in November of 2019 was to order an air incinerator, which, as I've spoken with experts on biocontainment, you know, would argue you really would only do that and take that step, because it's a pretty old technology, if you had some kind of HEPA filter failure, some sort of filtration problem.
And that was in 2019.
That was in November of 2019.
So you hypothesized that there was a HEPA filter failure at the WIV in 2019.
Well, to be clear, I'm not hypothesizing anything.
Okay.
What I have done is talk to experts who raise that possibility. You couple that with the fact that you have really top Chinese scientists, including the manager of the Wuhan Institute of Virology's BSL-4 lab, who in articles were really raising the alarm about what they felt were shortcomings in biosecurity within China, that there were not enough trained technicians, that there were budget shortfalls, and, you know, those create all kinds of risks. So it was the Chinese scientists themselves who were really expressing that in the strongest terms.
Susan, in 2020, you had an email exchange with other virologists that has now become, dare I say, has gone viral. Sorry, I had to slip that in somewhere. In which you were debating the correct name for the newly discovered virus. And the exchange read something like this from Sean Liu. But we cannot rule out the possibility of other bat viruses from the lab. The Wuhan lab has many bat samples not yet worked out or results published. There are some concerns that some of their samples may not have been handled properly and leaked out of the lab, but just a possibility. Your reply, do you think it could have come from a bat virus? Which one, or an unpublished one? RATG13 is the closest. Is it close enough in sequence? Do you think it came through an intermediate host and sequence drifted? This is a very chilling idea. Now,
Okay, so
can you explain why you said that?
yeah, it is a chilling idea that somebody would make a virus, but I don't think it happened. That's ridiculous.
I thought you were responding to the suggestion that it would have been a leak from
Okay, first of all,
a non-enhanced.
we are talking about the very, very beginning when nobody knew anything. RATG13 was identified. It's not even a virus, I want you to know. It's a bunch of sequences, so it's not even infectious. But it was the closest virus closest sequence to SARS-CoV-2, but yet it's still quite far away. Sergey may know the number of years away it is. But anyway, so that was all we knew then was that there was this RATG13 that looked like it could have been somewhat close to SARS. And the idea that anything that a bat virus infected a human and started this is a chilling idea. But I don't think there's any merit to it. And just let me say once more too that yes, viruses can leak out of labs, but you have to have the virus first before it can leak. You know, and these people Dr. Shi and others, they're colleagues of ours in the virology community. So I guess I have more just my inherent faith in them than you probably do.
Jamie, let you respond, and then I need to get back
Thank you for using
to
that word, faith.
Sergey.
Because it's just faith. We have no
just faith.
no, but we have no idea. So nobody, I mean, maybe a few people have said, but nobody's saying is RATG13 is the precursor to SARS-CoV-2. We do know that there were viruses collected in the same location.
It's not a virus even, it's sequences.
We know there were viruses collected from the same part of southern China where the sequences or the genetic information that informs RATG13 come from. That's the name of that virus. And again, we don't know what work was being done, but we know that they were collecting these bat coronaviruses. We know the type of work that they were doing. We know what the Chinese virologists were saying about the risk. We know that the French company that was partnering with China to build the Wuhan Institute of Virology refused to certify it because they were so disgusted with the safety levels of that lab. And so again, because there is no determinative evidence, and certainly I would love it if we just said, oh my God, here's genetic data or whatever, and it proves. We just have to apply logic in at least thinking about what is and isn't possible. And that's why I'm not saying I know it came from a research-related incident. I do not. But I do know, and I absolutely know, and I think it is, in my view, irrefutable that a research-related origin is a very, very, very serious possibility that must be actively investigated. And that's why I think we can all agree that it is just an outrage that China is blocking this kind of investigation and access to the data. And I'm sure Xu Zhengli is a wonderful person, but it is very different to collaborate with a scientist in New Zealand to a scientist working on an issue of this level of sensitivity inside of China. Xu Zhengli herself, people in these same U.S. right to emails are saying she was afraid that she was going to be arrested. Xu Zhengli can't just say, hey, there was a lab accident, are bad. I mean, we're dealing with an authoritarian state which, with complete control, Zhang Zhan is rotting in jail near death for asking questions about pandemic origin. So I'm sure.
So Jamie, you've teed that up perfectly for me, because the next thing that I wanted to talk about is lab safety. And I wanted to start with Sergey. So you grew up in the Soviet Union and Russia.
I did.
And you know what it's like to try to be a scientist and to conduct scientific research in a non-democratic environment controlled by a totalitarian government. Do you feel that, how much do you feel it is possible to separate the science from the politics? And how much do you feel that the government hovers over and controls the activities of the scientists, the information that comes out of the lab? How does that color your thinking about the data that you're working with in this area?
Well, that's a great question. Fortunately, I was just a teenager at the Soviet Union.
Yes, not quite as a
17.
full-grown scientist yet.
But going back to sort of an earlier salient point, I think one of the, so I have an antibody against sort of stifling scientific debate. And I think that's sort of the reason why the early, and I think significantly oversold insistence that we had this certain, almost 100% probability that one possibility was right and the other was wrong, never sat right with me even before I knew anything. It's just un-American, right? And I don't like it. I think people from my background that have experienced what, late-stage totalitarianism looked like have a very different perspective on, how people work and how to be suppressed. But yes, it is entirely possible. I mean, there's plenty of incidents in the Soviet Union of biosafety weapons cover-ups, Chernobyl, Mayak power plant explosion, all these things, anthrax leaks. It's just, history is littered with it. And the other thing that, I was kind of thinking about it is in terms of how accidents happen. Right? I mean, if you look at any industrial accident, I mean, the two things that came to mind, you think experts always do things right. I mean, go back to 1945 and 1946, Los Alamos Project. You had nuclear physicists that were very experienced with this, were fiddling with plutonium cores, and they just get careless. So 2 of them died within 6 months of each other because they irradiated themselves by violating safety protocols. I mean, we're talking about something that people knew about, right? Knew it was dangerous, and they still become inured to it. So they just kind of just work with things. And so it is very difficult, I think, to put a numerical probability of what is likely and unlikely in a complex laboratory setting for which you have incomplete information. So similarly, it is also very difficult to state what we can and cannot expect a virus to do. So Susan's absolutely right. Viruses are fascinating. We can talk about things that we certainly know. We've sequenced 60 million or more genomes from coronavirus, and I can tell you through stages that we've been surprised. When Omicron arose, where did it come from? Nobody thought about chronically infected patients, which is the most likely source, as a major vehicle for viral evolution. Nobody said it. Everybody thought Delta was going to be around. But just one example. We're not good at predicting things and evaluating relative probabilities of things going forward. And the final minor point I would like to make is, so, you know, there's this saying that the road to hell is paved with good intentions. So I would like to point out one scenario. You know, these diffuse projects, and I was tangentially involved in one that had nothing to do with this a while ago, they're trying to do a good thing. They're trying to predict, without trying to infect people, which animal viruses can potentially transmit. We know a lot of things. We know there are lots of viruses in animals. We also know that we know very little about them, tiny sampling of what's out there. We know that most viruses came from animals, effectively, you know, all of the clinically relevant ones. So the next question, how do you stop something before it happens? So you go out and sequence bats and you sample them and you get some sequences out of them. You know, I mean, you can go in and try to infect people, which is obviously unethical, and you never do this. You can do cell cultures, which would be expensive and perhaps not tell you what to do. What you want to do is you want to do data science. You want to do high throughput screening and say, this is the viral genome. I can predict with some degree of certainty if it's high pathogenicity or not. But we have no data about it. We have so little data about what's infectious, what's not. And what we do know from viruses is that sometimes minor changes make significant impact to what the virus does. You know, again, Omicron, alpha, beta, gamma variants, N501Y, all these things you've heard about. Single mutations, major difference. Okay. Now, there was an effort to collect data, or at least there was a thought to collect data like this. And how would you do this? You would potentially collect viruses and you would measure them in some sense about how infectious they are, with a purely benign goal of trying to identify what could but could not be potentially infectious. It's a huge upside. If you're able to do this, you just go and surveil all the things where animals could come from, you know, animal viruses could come from, you know, predict where you focus your efforts because you can't surveil everything. But you can also imagine how this benign and well-motivated example could lead to a scenario where accidentally people do things they don't expect to be dangerous. Just as Susan said, it's very difficult to make a virus, you know, pathogenic, and completely, this is not my space. I have nothing to say about you know, the lab complexity, but just think about what could happen in the other direction. Accidentally, you make a minor change and something big could happen.
So Gigi, you do have significant expertise on the topic of lab safety, standards, protocol, and so on. And one of the things that we've talked about is, you know, why don't governments and labs come clean when they do have an accident? And wouldn't that help us to improve lab safety standards? And so.
Sure. So first of all, biosafety is a very important topic. It's often a very underfunded area because, you know, you want to spend money on research, you want to spend money on stuff that helps you do research, right? So this is and it's an occupational health issue for sure. And nowhere is this more important than for people who are working in clinical labs. At least in research labs, like if the tube is labeled Ebola, you know there's Ebola in there. If you're working in a clinical lab in West Africa, you don't know what samples are going to come your way. And I've personally seen laboratory technicians in clinical labs in West Africa, take plates and smell them, because that is a good way to tell which kind of bacteria you've got in it, but it's not recommended anymore. And there have been big changes in laboratory safety over the decades, for sure. Even, just before my time, people used to mouth pipette. 60, 70, 80 years ago, people died of anthrax because they were working at a bench with anthrax, and then they got it on their hands, and then as they were smoking, they introduced it into their lungs. That doesn't happen anymore. But so biosafety, why is it important, and why can't people admit to it? Well, this is why when you're designing a biosafety program, you have to have a lot of no-fault conditions in place, because people do not, if it's the institutional policy that you're not supposed to do a certain thing, you did a certain thing and you get sick, then you're not going to want to come clean about it. So that's why it's biosafety is really important to have a program that's in place that encourages people to report, where you monitor people's health and everything like that. One of the things about we can speculate a lot about the Chinese labs. I've visited some, not the WIV, but we can speculate that they had more or less safety considerations. I certainly haven't seen any direct evidence that there was less safety considerations, and some of the scientists who visited those labs and then went elsewhere said it was just like, working in any other lab. But all those things can be true, and I just want to introduce you to the term from the intelligence world, which is mirror imaging. And that is, sometimes you bring your own preconceptions to what is going on, and you make conclusions based on what you would be doing if you were in that situation. You're looking in the mirror, you're looking at your own motivations. And so, it could be that there's a different explanation, and it's been a real challenge in the intelligence world, why did Afghanistan fall very quickly? Why, maybe thinking maybe the World Trade Center wasn't going to be attacked again, or, things like that. Maybe the Soviet Union is going to be stable for another few decades. These are things that people have posited that have been wrong based on not looking to enough experts, not thinking enough of what the cultural context is of what's going on. So I think there's a lot, I'm sure that every lab can improve in biosafety. And we could spend more money on biosafety. I'd love to see programs in biosafety. I'd love to see more biosafety officers. I'd like to see people get PhDs in biosafety. That's all great. But we don't know we just don't know that has anything to do with the collection of evidence, all the epidemiology. And if there was a lab accident, it would have to then fit with the data that we have. You'd have to have a progenitor virus making people sick, and then they had their book club at the market. It would have to make sense
And be able to trace
to be able to fit the epidemiology.
Thank you. I'm going to let the audience in just a couple minutes. We're going to wrap up with a couple of last, much broader questions. The first is then really comes out of this discussion, regardless of what side of the origins debate you're on. We've all expressed some concern about the lack of transparency of the Chinese. We've also expressed some concerns about the lack of transparency of our own intelligence community and the need to have the data from our own government about why the intelligence communities are reaching the assessments they are. I think we're all waiting for the declassification of this intel, which may or may not generate much information. But I want to put this in perspective as someone who is a scholar of war for you, and who works with the Army a fair bit. In the bowels of Pentagon, experts are studying what they regard as the biggest security threat right now, which is China. And they're preparing for the possibility, for which the Russian invasion of Ukraine appears to be just a warm-up, that China will invade Taiwan. And they're putting this on roughly a 2-year time frame. So I'm, saying this so everybody can kind of get real. It may or may not happen, but this is where the thinking is, and it's quite nonpartisan thinking about the military risk that China currently poses. You may have seen a very little story that Japan, which is much more attuned to this risk than we here in the United States are, has been taking already a series of countermeasures against this possibility, and China is already taking countermeasures by threatening the Japan with regard to certain Japanese islands. So we're preparing and having initial stages around a major world standoff around the status of Taiwan. The U.S. is more or less pledged to defend Taiwanese independence, but even if the U.S. would decide not to defend Taiwan militarily, and it was more of a Ukraine-type situation where the U.S. decided to withdraw economic support and try to disentangle economically from China, forecasts and war games type analysis show that we would very quickly grind the U.S. economy to a halt. Now, this is very important because at the same time, we have a robust community of scientific collaboration between the U.S. and China, just as we have a robust community, economic interchange that absolutely dwarfs the kind of economic interchange that we have with, or had with Russia. You know how difficult it has been for Western Europe to begin to disentangle and make themselves less dependent on Russian energy sources. Magnify that vastly more when you start thinking about disentangling economically from China. I put this in a context because I think it's important for us to think about this context when we start thinking about these scientific collaborations. It's very important for the scientists to say, hey, we need to be able to work with our Chinese counterparts. We need to be able to work in these Chinese labs. We don't want too much interference. We don't want international bodies telling us what research we can do and what we can't do. We don't want the US government telling us, shutting us down or on some spooky theories of spying, telling us that we can't do important gain-of-function research. But there is a larger context here. So should this research be done with China or not? And if so, what are the precautions that should be put in place? Okay, Jamie.
Thank you. So you're absolutely right, and I really appreciate what you said. There is an irony of this moment in history that our fate is interconnected with not just China, but with the rest of the world. And there was certainly an idea that began in the late 1970s with Deng Xiaoping and the opening up and the reforms. There was an idea, well, let's weave China into the rest of the world, and then China will go the way of Korea or Japan or any other number of other countries that were part of this fabric. And so that's the world that we live in. And then after the first SARS, there was the idea, well, this was a really scary moment for the United States because SARS-1 and other pandemics, they start in other parts of the world, generally tropical Asia and Africa. And we realized that we didn't have the capacity in those other, we collectively as humans, in those other places. So we needed eyes and ears in those other places. We needed scientists like Shuzheng Li and others, and that's why the US government invested in training those scientists. We needed facilities so that samples could be analyzed there and not everything wasn't coming to here. And then we have many, many wonderful people who have dedicated their lives on this idea that the way that we get to a safer, better future is by deepening collaboration between lots of countries, but the United States and China. And science felt like a great way to do it, and it is. And it's really just wonderful. But I was telling Claire before this, just imagine that you are on a boat on Calypso and you're with Jacques Cousteau. And Jacques Cousteau is 80 years old, and you say, Jacques, congratulations for all the wonderful work that you've done. I know how committed you are, but hey, I just need to tell you this. It turns out there's a small leak in your scuba gear, and all these reefs that you've been visiting, there's been a little leak and a bacteria has escaped, and all those reefs are now dead, and all those animals are now dead. And he would say, no, c'est impossible. And that's this moment that we're in, and that's why, in my view, there was an instinctive immunological, that's not respectful.
Sorry.
Yeah, that's not respectful. And that's why there was an instinctive immunological response for people who've dedicated their entire lives to this one very noble idea that building these relationships is the foundation of this better world, to even considering the possibility, whether it's correct or not, even considering the possibility that this very well-intentioned thing could have played some contributing role to this very devastating thing. And that's why, as I said in the beginning, I'm a Democrat, a liberal, a progressive, all the things that I imagine I share with all of you. But I think that we can't be guided by the purity of our intentions. We just need to look at all of the available evidence. And in my mind, that available evidence points to the very real possibility of a research-related origin. And it's going to force us, unfortunately, sadly, tragically, to evaluate our scientific collaborations with China in a deeper way, because right now, when COVID happened and we wanted to know, well, what was the research that was happening at the Wuhan Institute of Virology with a small amount of United States government funding, we weren't able to get that. When we said, well, how about the database of of their viral samples that the United States may have played a small role in funding, we didn't have access to that. And so I just think that we're going to have to evaluate our relationships based on common values and common principles and common behaviors. And unfortunately, I think that's why we're seeing, maybe it's not a decoupling, but I think an active de-risking in the United States in our relationship with China.
Yes, please.
So, I think that science has always been a way for nations where there's tensions, to collaborate. And that was very true with the Soviet Union. And that was very true after the dissolution of the Soviet Union. And that the US invested very heavily in labs around the world to, one, redirect Russian scientists and Soviet scientists, but also to raise the level of public health. I don't think that it's a pure, we can't see that science could be misused. I was personally on the inspection team that went to Dugway after, which is the Army base, which I don't know if you remember, this is now like 8 years ago, when they accidentally sent out live anthrax samples all over the world. It was the same strain as used in the anthrax letter attacks in 2001, so it was really awkward, to say the least. Nobody was hurt, but it was very embarrassing, and the Army was absolutely at fault with that. And that was the result of our inspection. I was on the inspection team for the FDA cold refrigerating room, the cold room, thank you. Sorry, I need a little more coffee, where smallpox was found. And, it was obviously an error that you shouldn't let like a cardboard box sit there for 50 years without somebody checking to see what's in it. So, I think we're able to point out when things are wrong when we see it. It's just that the balance of evidence is on this being a natural emergence of like really bad behavior of the Chinese. It's not like the wild animal market is a good thing. It's a terrible thing. It's an illegal practice. There's a ton of money in it. It's going to be very hard to turn that off. The amount of illegal activity in China alone and the illegal wildlife sales rivals the U.S. beef industry. That is a lot of money, and so it's really hard to course correct. But as far as what you were getting to is norms development, and if we don't collaborate with the other big scientific powerhouse in the world, if we don't work with them in a productive diplomatic way, then we are ceding control, because they will pour money into this and they will make decisions. Okay, these are the biosafety steps we need to do before we release a gene drive to kill mosquitoes in this area. Maybe they would take fewer steps in biosafety than we would. But we're not going to be able to influence that if we're not working with them.
But that's already happened. I mean, that, in my mind, is the most likely story of what we're experiencing now.
I need to get the audience in, which has been very patient. The last question for this group is, who should be at the table in making these decisions? I sense and have heard from many scientists that the issues to be resolved here are purely scientific issues. Catherine, I'm going to give a last question to you. Why should journalists be part of this world? Should journalists keep reporting on this stuff? I know it's been personally very challenging for you to be reporting in this extremely contentious landscape. Can you say something about, do you intend to keep reporting in this area? And do you think journalists need to keep working in this space?
Well, let me just say that every journalist who is reporting on COVID origins that I've talked to, they all say the same thing, which is this is the hardest, most complicated story they've ever reported. It is just, and that's the case for many reasons. There is just information we don't have that we may never have, right? So it is an unresolved question, and tempers have flared, and emotions are running high. Our own government has not been forthcoming really across the board. We don't know what the intelligence agencies know. There is going to be a declassification that we are waiting for, but we don't know how much will be declassified. Intelligence agencies have come to differing conclusions. We don't know really the how or the why. And there's also all kinds of information that we don't have and we may never have from the market, as Gigi talked about with the discarding and taking away of animals. So, as a journalist, you like to work on a story where you can answer fundamental questions. And it is incredibly frustrating for journalists to be, killing themselves on a story where the answers may never be forthcoming. So it's extremely challenging. Some folks have made the argument that really only scientists should be investigating this. That I strongly disagree with. I think that, because of the efforts of journalists and because of the efforts of FOIA groups, we've uncovered a huge amount of information from emails that really never would have seen the light of day. And one of the things that those disclosures told us is that many scientists involved in this debate have considered the possibility of a lab leak. They didn't necessarily think it was a crazy conspiracy. They thought it was a possibility. So, I think it is critical for journalists to keep chipping away at these questions on both sides. Certainly I hope that more information of all kinds will emerge.
Great. All right. The floor is finally open. Members, I'm sorry, Gigi, I'm going to grab this. Members of our audience, if you wouldn't mind just stating your name and letting us know who you are. Sir. And do we have a microphone coming around for you?
Testing.
Hopefully it will work.
Okay, he's very innovative.
Hello? Hello?
Yes,
Hello?
we hear you.
So thank you very much. My name is Rick Bushman. I'm the chair of the microbiology department at the Perelman School of Medicine. I've published a bunch on SARS-CoV-2 with Susan.
And indeed, we're just quoted in the New York Times.
Yeah, associated with the raccoon dog thing. So a couple of points. Penn professor Jim Allwine has an article that just came out with several other virologists that presents, I think, the common sense view step by step. So I'd urge you all to look at that for background. One thing they pointed to, and some of you have mentioned, there have been 9 jumps of coronaviruses from animals into humans. And so here we have SARS-CoV-2. What do you think the answer is? It seems to me that this is very much the default hypothesis until there's some evidence going the other way. And we've not seen evidence going the other way. So, maybe that'll change tomorrow. I'll change my view. But the zoonotic idea is so well established, and there's no evidence for the other idea. With regard to the raccoon dogs, what Mr. Metzel said was wrong. The New York Times did not recant their view. So just to give you a little background, so they're going around swabbing the seafood market. They're testing sewage. So these are low quality samples. The material's degraded. Maybe it's been there for a while. There's somebody cut up some fish and then coughed, and so you find fish sequences and SARS coronavirus sequences there too. The data's all low quality, so much turns on exactly how you filter it, what you do with it. So what thresholds do you apply? So the first paper was Warraby and a group of Americans. They found associations of SARS-CoV-2 with a number of different animals, including a strongly infected, a sample with a lot of raccoon dog and a lot of SARS-CoV-2. So maybe that was an infected raccoon dog. The Chinese then published their paper, and they saw sort of data in the same direction, except they couldn't place the raccoon dog fully accurately, so they called it Canis. So the name of the species changed in their figure versus the American study. And then came the Jesse Bloom study. Jesse applied a quite stringent filter to this very low quality data, and some of it went away. And so he got a pretty different answer. But it's all about the data being low quality and then people choosing thresholds. So what I was quoted in the New York Times as saying is usually the careful way to do it is try a bunch of different thresholds and see how that affects your conclusions. And in this case, I think you would find it probably did have pretty strong effect on your conclusions. So you can see in that the first couple of papers that there's association of SARS-CoV-2 with raccoon dogs. So that kind of looked like that's weak evidence for an infected raccoon dog. But how did that happen? Did a human infect a raccoon dog? Did the raccoon dog infect a human? As people have rightly pointed out, it's like a month after, at least, that you think the epidemic started. So that's not the raccoon dog that infected people. But probably there is evidence. I take that as evidence, weak evidence, for circulation of the virus in animals in the market. And so that's kind of a little bit in the direction of the zoonotic jump, but not very strongly. And so I think in both of the, for the zoonosis or lab leak, there's the question of how did the virus get to Wuhan? I've talked about this with Jesse, a point he makes. So it could be that it was in some animal, or it could be that someone from the lab was in a bat cave and collecting samples and they got infected and they came back and coughed on someone in the market, and that's how it started. So, I mean, that's not ruled out. It's just there's no evidence for it, and we have all these examples of zoonotic infections and viruses. So, I mean, it's pretty hard to not have that be the default view, given the very extensive history and epidemiology. So I'll stop there.
Thank you very much. Okay, so I have a bunch of hands in the air. Let me get Nick Evans, whose area of research this is. And Nick, if you can just introduce yourself.
Thanks, Jen. Nicholas Evans, I'm an associate professor at the University of Massachusetts Lowell, and as Claire said, I've been thinking about gain of function for a while. Hi, Gigi.
It's so good to see you.
Good to see you too. It's like a pandemic, like kind of reunion. This is great, right? And yes, my hair has gone from short to long to short again, which is very weird. So I do have a question about the stakes of this debate, and I think that this is kind of the thing that I'm sitting here for the last hour and a half thinking about, because people are talking about what they know and don't know, and just so we're not all committing faith moves, as been said before. Knowing X and knowing an inference to Y from X are two different things, right? So like just knowing a fact doesn't mean you actually know anything about how to infer to the next thing in that fact. But my big question here is there are two kinds of stakes that we could ask. One is, what are the stakes of answering this question now, given that the SARS one, origin story took 10 years to solve, and we're only at year 3? And 2, what are the stakes of answering this determinatively in any way, shape, or form, given that something like 60% of biodiversity in this country, in this world, is located in Southeast Asia, and we're probably going to have to deal with these countries for a lot longer, just because that's where those viruses come from. Because it's not clear to me, like, yeah, all right, maybe we get to a lab leak. Like, I mean, you know, like, I'm actually in the it doesn't matter camp, because I'm really controversial. But even if we answer a lab leak at the end of the day, what does that actually do for us, right? I mean, does it do anything? I don't know, and I would really like an answer from the good people on this panel about that.
Very good. Who wants to take that? Jamie?
Yeah, I mean, I've spoken so much, and Sergey's spoken so little. Can I go after Sergey?
Sure. That's a great question. I've thought about it a lot, and I don't have a good answer for it, because I think your stakes will depend on how you view this. I think Dr. Bushman made an excellent point, which is where we are really now is in, you know, statistical terminology. You're falling back on your priors. Everybody has some belief about how things are likely or not likely, and I would argue that there's very little data at the moment to move your priors. The same thing about the effect. I mean, obviously, I mean, just from a human perspective, some people might want some closure, right? I mean, this has been a very disruptive event. It would help you to know, you know, how it happened, because that would, you know, tint your perspective, so to speak. But I really like your first point, and I think I would like to emphasize that, which is science takes time to develop. You need more data, more scrutiny, more adversarial evaluation of data, as we see here. And demanding that we have an answer now is both impractical and I would say probably, I mean, in some sense, unethical. Because we know as more, I can give you examples of data which will completely change this around. If there's an infected animal, that's a big point in that direction, you know, of the market spillover. If there's evidence of cases outside the time window that we now think the first cases emerged. If we find a case in November in the United States, hypothetically speaking, flips everything upside down, we just throw it out the window. You know, so we're in a fragile state. The entrance of new data is likely to modify, significant new data is likely to modify the way we view this. So, you know, basically making any deterministic statements that this is more likely or less likely, you know, I don't think I would be comfortable with that at this point. But I think, you know, people with more global and political connections can speak to what this means in terms of, say, our geopolitics with China, because, I mean, say, yeah, so what would happen if, you know, we did decide it came from a lab? I mean, what are the ramifications of this? Do we want to know?
Yeah, so maybe I'll jump in just very quickly. I agree with you, Dr. Bushman, that for the market, based on the evidence that we have, we just don't know. And I think that's a very valid thing. And so then the question is, as Sergey said, well, what are the priors? And one prior is that most of these jumps have been zoonotic. And then you have to say, well, how much do I value that it happened in Wuhan? And so I think that's why there's just a lot of whether it's bias, priors, just how we value different types of arguments. But I think it's very valid questions. And then the stakes question, so to Sergey, what would happen if we found out tomorrow that it actually comes from a lab? Well, right now, what we've learned is, let's just say it comes from a lab. That means we live in a world where regulators in Uzbekistan and independent scientists in Uzbekistan are making decisions that will determine whether 20 million, 10 billion, actually we don't have 10 billion, 1 billion people live or die. And it's just insane that we live in a world where the capacity to have this kind of global reach is so decentralized and we don't have global governance systems. So if we found this out, I think that the efforts to have a global system for biosafety and biosecurity would get a huge boost. In 2005, after SARS 1, there was a renegotiation of the international health regulations. And the basic problems, the same as climate change, is we live in a world where global problems cannot be solved. And so now they're being renegotiated again, and I can guarantee you that it will be next to worthless, just like the IHR was totally worthless in the start of here. And so unless we can just say, hey, what's the problem, and identify it, we're just going to keep floating into a really dangerous thing. And so that's why if I had a magic wand, I'd say I hope this comes from zoonosis, because it brings us together. That's the desirable outcome.
Well, everybody seems to want to comment on this. Gigi and then Catherine.
So I think we have to be open for the possibility that this takes time, as you know, that it will, we'll have more information at one way or another. The information that emerges has to, or what we think has to fit all the data that we have. But what I think is really dismaying about a lot of the lab leak speculation is that, as you know, One Health is something that nobody disagrees with. We all know that we are connected to animals in our environment, but actually making productive change and doing something so that we're no longer encroaching on their territory, that we're preventing spillover risks, that we are taking on this tremendously moneyed bunch of issues, that that's not going to happen without the clear, I think, determination of the origin. I think and I worry that it's going to be SARS-3 or SARS-4 before we realize that we can't just take the property of bats away from them and live next to them and expect there not to be any consequences.
Just from a journalistic perspective, this story of origins, excuse me, has been on a kind of pendulum that just swings back and forth. So, excuse me, we went through the Department of Energy changes its intelligence assessment news cycle that went on for a couple of weeks. That was followed then by the raccoon dog news cycle that went on for a couple of weeks. And I think one issue for journalists is that the journalistic news cycle is much quicker than the scientific, research cycle. There's really a fundamental mismatch. Science takes time. And one thing that we've seen as journalists that's been striking in this pandemic is that, in a faraway land long ago, journalists would wait and cover peer-reviewed papers. And then, over the time pressure and coverage of the pandemic, journalists started covering preprints. And one thing that was striking with the raccoon dog story is at the point that burst into public view, there wasn't even a preprint. So it just,
Now they covered Twitter comments.
yeah. So it's just, it's gotten quicker and quicker and quicker. And while journalists are sort of chasing this story and waiting for some kind of resolution, I 100% agree with what Gigi said, that the world is not making much progress. The wildlife trade continues, and you see a proliferation of laboratories around the world, and also in some countries where regulation is less than stellar. So it seems that the search for, the efforts at any possible solution, regardless of the origin, sort of stalled behind this debate. And it would be great to make progress on some of the policy issues.
So we started a few minutes late. We should end soon, but I saw a couple of hands in the air. Let me take 2 questions at a time. We'll have a quick round of answers and then we'll close. David.
David Harrison, I'm a law school graduate. Speaking as a lawyer, it seems to me that the China's withholding of documentation raises a negative inference and seems to speak as evidence of a leak. I don't think anyone's mentioned it, but I had read that the pool of bat viruses is in South China, about 800 miles from Wuhan, and I think Sergey mentioned that the Chinese had been getting bat samples from them, and yet you would think that there would have been some evidence of the illness back in the bat, where the bats were, and not 800 miles away in Wuhan. I wonder if there's any comment on that.
Great question.
I say the animals could have been infected somewhere else too and brought to Wuhan.
Yeah, but then
we don't
there
know
would
where
be
they came
some
from.
trace if they were coming from animal farms. There were just Wuhan is a very sophisticated, wealthy city, a minimal trade in wild animals compared to so many other cities in China. You would see some evidence of it.
But viruses spread in dense populations, not out in the
There are
countryside.
lots of dense populations in China.
Well, yeah, it just happened in Wuhan. I mean, I don't think that's so mysterious.
So, David, you sparked much discussion with your question. I can see that we're going to have to reconvene this group for further discussions. It is my hope, in fact, that this will be a continuing research project for Searle. I think there's much for us to do. What's important is this is exactly the kind of interdisciplinary project that the Center for Ethics and the Rule of Law, and now in partnership with the Annenberg Public Policy Center, works on. It combines not only the important scientific questions that we've been discussing, but critical questions in national security, the rule of law, ethics, and of course global questions around international cooperation and lots of things we didn't talk about, like the possibility of a pandemic treaty and preventing the next pandemic. I will also say we bring these programs, many programs a year. I hope if you came out just for this one, you will look at some of our other programs. We have a book talk series. We have regular programs in the area at the intersection of ethics, military topics, topics relating to national security, bioethics, and other areas, cross-disciplinary issues on a regular basis. You can join our mailing list. We also never charge admission for our programs, and therefore we rely entirely on donations to sustain the work that we do. So we very much ask you to consider making a donation to our programs. There is a QR code that you can use that is located, where would we find that? At that back table, I think. And that links to a credit card link, and we'd be very grateful if you're interested in becoming involved in the work of the center. And for anyone here who is a student in the audience who's interested in future years in participating in our student summer internship program, please speak to our executive director, David Johnson, or in engaging with us during the year, because we have ongoing research that we do, all interdisciplinary research where we take law students, graduate students, even some undergraduates, to bring them together to do interdisciplinary research on topics at the intersection of these different areas. So thanks to our very, very sophisticated, interesting, and patient panelists for engaging in this complicated and difficult discussions, and to all of you, please let's give them a big round of applause.